everybody. So I have met Elizabeth once a couple of years ago during her visit in Göttingen at our institute, and we spent almost, I think, 2 hours in my office and discussing science, and in particular the possibilities to use perhaps fluorescence cross-correlation spectroscopy measurements to investigate some aspects of the dynamics of the spliceosome. It was a great brainstorming meeting, and we discussed possible collaborations. Unfortunately, at that time we were not ready for applying these methods because only recently we could reconstitute spliceosomes from purified components, and indeed now we have applied FCCS, and I will show some data on this, and I'm sure Ellie would have liked these kind of applications. So I have a very good memory of Ellie in this way. So I'd like now to first thank the organizers for inviting me to this wonderful meeting, and I would say this event here in Buenos Aires. In fact, it's the second time I'm here in Buenos Aires, and I feel reasonably well connected already to this place, because you have a great splicing community here in Buenos Aires with Alberto Kornblit and Annabella Threbrov around. Annabella is, I think, in the audience. Alberto, unfortunately, could not come because he's just being knighted by the American Academy of Science. And while Alberto would have talked to you about co-transcriptional splicing and the regulation of alternative splicing, my group is mainly interested in the elucidation of the inner workings of the core splicing machinery, and we are using a multi-parameter experimental approach, including biochemistry, biophysical methods, and structural biology, to dissect this machine, which is responsible for the removal of introns from nuclear pre-mRNAs, and which I think is one of the most complex molecular machines in eukaryotic cells. I would like to start out pretty simple to give you some background to the spliceosome, and in fact the chemical pathway of pre-mRNA splicing is indeed very simple. It proceeds via 2 phosphoester transfer reactions and via 2 intermediates, the free exon 1 and the exon 2 intron lariat. And due to the fact that it has only short cis-acting consensus signal sequences which are around the chemical reactive sites, the 5' splice site, the branch site, and the 3' splice site, nuclear pre-mRNA introns cannot remove themselves by a self-splicing process out of the messenger RNA, but rather need myriads of trans-acting factors which make up the spliceosome. And one essential family of trans-acting splicing factors are the so-called small nuclear RNPs. We distinguish the U1 SNRNP, the U2, the U4, U6, U5 RNPs. They contain either 1 RNA or 2, like the U4, U6 diSNRP, where these 2 RNAs are base paired together, and under splicing conditions, U4, U6 diSNRP and the U5 particle form a U4, U6, U5 tri-SNRNP complex. These, in addition to the RNAs, they contain many proteins, altogether about 50, which make up already for about 3 megadalton of the molecular weight of the spliceosome. The next slide shows you the assembly pathway of the spliceosome, and it shows already an interesting design principle of this molecular machine, because the spliceosome does not exist on its own in the absence of the substrate. Instead, it assembles de novo on each intron from scratch in a stepwise manner, and here I focus just on the assembly of the SNRNPs and a group of 8 evolutionarily conserved Rna Helicases, which are also essential components of the splicing machineries in all eukaryotes. So it starts in the early stage by recruiting U1 and U2 to the 5' splice site and the branch site, yielding the A complex, followed by the recruitment of the tri-SNRP to give now the B complex. This is a multi-megadalton ensemble which doesn't have an active site, another design principle of this machine. The catalytic center is now created in a stepwise manner, and in the early stage, due to the action of BER2, U5 resident RNA helicase. U1 and U4 are displaced. We get the activated complex. This needs to be catalytically activated now by another RNA helicase, the PRP2. And now this complex can catalyze the first step of the splicing reaction, yielding the C complex, which comprises both intermediates of the pre-mRNA. Now the catalytic center has to be remodeled by the next RNA helicase, PRP16, to make it ready to catalyze the second step. We get the mRNA product, and this has to be released from the intron-lariat spliceosome by the RNA helicase PRP22, and this intron-lariat spliceosome is then disassembled, and the particles are used for a fresh round of splicing again. Now, due to these design principles, this spliceosome can be a nightmare for people like us who are interested in the biochemical analysis and in the high-resolution structure determination, because unlike the situation with ribosomes, here you cannot just grind up the cells and purify endogenous spliceosomes, because you would end up with a very heterogeneous population. What instead we have to do is to add a model pre-messenger RNA to a splicing active extract, and we use yeast and HeLa cells as model systems. And then you have to invent methods to let it assemble to certain stages of the spliceosome assembly pathway, and then to purify these snapshots. That means we have to purify all these snapshots and analyze them one by one. And so we have done this over the last couple of years, and the first question we answered was how many proteins are involved in the splicing machineries of the human cells and yeast. And the number is 170 for the human splicing machinery, and yeast does it with almost half of them, with about 90 proteins. And the interesting observation here was, in the first place, that except for a handful of these 90 proteins of the yeast spliceosome, all had functional counterparts in the human spliceosome, which means that the yeast spliceosome apparently is an evolutionarily conserved core structure which has all the capacities of the basic splicing machinery, and the catalytic mechanisms are essentially conserved between human and higher eukaryotes. Now, what I would like to do now is to focus more or less just on the catalytic activation of the spliceosome, how this pre-catalytic B complex is transformed into a catalytically active machinery, and then to talk about the catalytic phase of the spliceosome, the remodeling, the structural dynamics. I will also talk about the effect of these RNA helicases on the structure of the spliceosomes, and at the end, I will show you some cryo-EM structures of some human spliceosomal complexes. Now, when I say structural dynamics, I mean the following. And if we just look at the protein composition and compare the B complex with the activated complex, then you can see that in the B complex we have mainly the SNRP proteins and another structural module, the PRP19 complex, constituted of about 10 proteins. And during the transformation to the activated complex, what happens is that not only U1 RNP proteins are dissociated, but all of the U4, U6, and most of the tri-SNRP proteins are displaced. About 35 proteins are leaving the spliceosome. On the other hand, about 10 proteins are now stably and stoichiometrically integrated into the activated spliceosome. So the signature between these two snapshots is dramatically different. At the same time, dramatic remodeling occurs at the RNA-RNA interaction network in the spliceosome. So in the B complex, this is the exon-intron-exon structure. In the B complex, U1 is base paired to the 5' splice site, U2 to the branch site, and the U4 and U6 are still base paired together. Now the activation happens, and in this case, U4 is dissociated from U6 RNA, U1 is dissociated from the 5' splice site, U6 rearranges to form this intramolecular stem loop, base pairs with U2 and with the 5' end of the intron. And what we are looking at here are structural RNA elements which are at the heart of the catalytic center. It is basically a magnesium RNA-based catalytic mechanism. However, nothing works without proteins, and the proteins have to shape the catalytically active conformation of the RNA, and they also help to position the pre-mRNA splice sites into the active site. So it is in fact a ribonucleoprotein enzyme. So what I'd like to discuss now is an important RNA helicase which acts by dissociating the U4 RNA from U6 RNA, and this is called BRR1 or BER2, and we have shown that it is loaded onto the single strand of the U4 RNA and then displaces in a 3' to 5' direction the U4 RNA. And this BER2 protein has several interesting features. First of all, it is a permanent resident and the only RNA helicase of the 8 spliceosomal ones. It is a permanent resident of the spliceosome, and it coexists with its substrate, the 4U6 RNA, in the trisnerp, and therefore it has to be tightly regulated in order to prevent premature activation. And moreover, it is the largest helicase in the spliceosome with about 250 kilodaltons. And it is the only SCI2-like member of the SF2 family of RNA helicases, and moreover it has a dual cassette organization. So the amino terminal cassette contains these RecA domains followed by a Sec63 unit, and this is duplicated in BER2. So in a collaboration with Markus Wahl, we set out to crystallize this protein, which didn't work with a full-length protein, but fortunately we were able to get a stable 200 kilodalton sized fragment by proteolytic digestion, which fortunately comprised both helicassette units, and this crystallized readily, and this could be solved at about 2.65 angstrom resolution. And this is now the structure of BER2. This is the amino terminal helicase cassette, and this is the C-terminal helicase cassette, and you can see immediately how tightly they are packed against each other. It is about 50 nanometers long and about 10 nanometers in width, and it immediately tells us that it apparently functions as a functional unit. If we turn it around by 150 degrees, we are now looking into the amino terminal cassette, and and what we had shown previously is that only the amino terminal cassette apparently is active, though the C-terminal cassette is also required in yeast. If you delete this, yeast is no longer viable. And we have shown that this is a hub for protein-protein interaction with many spliceosomal proteins. And we are looking now here into the RNA binding channel of the amino terminal cassette, and there is a circular arrangement of certain structural modules. Here are the 2 RecA helicase domains, followed by a wing helix domain, a helical bundle, and a helix loop helix domain. And this is an IG-like fold which belongs to the amino terminal cassette. Both cassettes are structurally very similar. And so this is the circular arrangement here is typical for these SF2 type helicases. And what is also important is this hairpin loop here. It is called a separator loop. And the RNA we have modeled in the U4, U6 RNA into this binding tunnel here. And this long ratchet helix here drags the RNA in a 3', 5' direction across this separator loop, and this wedges into the double strands and opens it up. And by mutational analysis, we could support this model that U4 RNA is located in the RNA binding tunnel in this way. For example, this, point mutations of the separator loop kill the enzyme, and mutations in this ratchet helix again also downregulate the RNA unwinding activity. And the exit here across the helix loop helix is documented by point mutations in this region here, which shows that then there is no unwinding activity, so that we can be confident that U4, U6 RNA is unwound in a 3', 5' direction by the amino terminal cassette. So why then do we need the C-terminal cassette if it is not active on its own? A hint to this came from a comparison which we did by just using the amino terminal cassette and comparing the unwinding activity of U4, U6 with the entire helicase region, and this shows how dramatically different the activity is, which suggests that the tight packing of the C-terminal cassette, though inactive on its own to the amino terminal cassette, activates now this cassette. And this was further supported by mutating several residues along this communication line, and this always downregulated the activity of the amino terminal cassette. And this is consistent with our previous observation that proteins may interact with the C-terminal cassette in this way, could regulate by communication via this communication line the activity of the amino terminal cassette, and only recently we could show that another U5 protein, PRP8, can shut off by binding to the BER2 entirely the RNA helicase activity, which is probably important to prevent premature opening of U4, U6, and we have now a co-crystal and are currently analyzing the structure, and it will be interesting how BER2 downregulates the helicase activity and prevents RNA binding to this cassette. I'm now switching gears and would like to talk about the implementation of a reconstitution system from purified components, which allowed us now to recapitulate in the yeast pre-mRNA splicing system both catalytic steps and the catalytic activation. Until recently, it was only possible to analyze the function of a certain splicing factor by immunodepleting this from splicing extracts, then go for the biochemical phenotype, and then add back recombinant and mutated proteins. And now we have established a reconstitution system. Let me just illustrate how we established this. And here we are making use of a yeast strain which comprises a temperature-sensitive mutant of the PRP2 RNA helicase. And this is the helicase which catalytically activates the activated complex. And so what we do, we use the heat-inactivated extracts from this mutant strain and then allow, by adding a wild-type precursor RNA into the extract, the assembly up to the activated complex. We purify this and now add back recombinantly produced proteins, for example, the PRP2, SPP2. This is a co-activator of PRP2. And then we asked which factors are required and are necessary and sufficient to now recapitulate the first catalytic step and the second catalytic step. And these two proteins recruited gained our attention because CWC25 and 23 were always co-isolated with the C complex, which we previously purified from splicing extracts, and there were no functions assigned to these proteins, and they could be splicing factors. Moreover, these factors were already known from genetic experiments to play a role in the second step of splicing reaction. However, their exact function was not clear. PRP16 is the RNA helicase, which is essential for the second step. So this shows you now the only or one of the few RNA gels, splicing gels I want to show you. This is a denaturing RNA gel. This is the precursor RNA, which is uncleaved here. This is a degradation product. Don't pay attention to this. And if we incubate now the purified activated complex with ATP, there is no first step of splicing to be seen. Only if we add PRP2 RNA helicase and its co-activator SPP2 together with ATP, we see a minor amount of intermediates. This is the exon 2 intron lariat. So this tells us that indeed we have catalytically activated the system. However, we need something else for an efficient first step of splicing. And indeed, it is the CWC25 here in the second but last lane, which then yields a very efficient step 1 catalysis of about 70% yield. And in this way, we discovered a new splicing factor, which is essential for the first step of the reaction. It is an interesting protein. It doesn't have a motif. You can boil it for half an hour.